Hello students and welcome to my Unit 6 video on the chemistry of life. This video will help you study water, the major biological macromolecules of carbs, lipids, proteins, nucleic acids, and enzymes, which are proteins as well. Like all my videos, this is not for profit. It's just meant to help students out there. I use lots of images from Khan Academy. You can check that out on khanacademy.org. I also encourage you to check out your free textbook, CK12, online as well. This picture up here is Wadi Dam. I got to go up there near Jebel Shams in Oman during the Eid break in it. It was just beautiful, my goodness. And then the water was running, and you could see it's just promoting life here. And it just made me think about all the inherent beauty that we have in this country and how it's all founded on biology. So let's dive in. This is me and my daughter hiking to that spot. Couple quick video tips. Stop this video off and ask yourself questions, construct your own explanations. Don't just memorize what I say. All right, you should be working on trying to put it in your own words, crafting your own explanations, going off on your own investigations. That's the beauty of NGSS. All right, here are some questions from the study guide. What is an atom? What's the octet rule? Can you draw covalent bonds that can make water? Can you label the polarity of water? Can you draw the bonds between water molecules? And can you describe how hydrogen bonding leads to emergent properties that contribute to life? If you could do 1 through 6 here, then you can skip this part of the video. There's no point to watching it. If you can't, then let's dive in and get you there, okay? I told you that there will be nothing on the periodic table or atoms or elements. There will be no chemistry on this assessment. But it's helpful to know a little bit as we try and understand water. Here is a periodic table. I'm sure you've seen it before. And you have elements on here, right? And elements can't be broken down into any other substances. They are the most basic part there. So an element would be hydrogen. Another element could be oxygen, right? And if you combine 2 hydrogens and an oxygen, then you get a compound that's a mixture of 2 elements, and that makes water. You might combine Na and Cl to make table salt, or carbon and 2 oxygens to make carbon dioxide. So an atom is the smallest particle of an element. And then when they come together, when multiple elements come together, they form bonds. So you can have a carbon atom. You can have a helium atom. And here is a picture of a helium atom. Helium is number 2 on the periodic table. It's going to have 2 protons, 2 neutrons, and 2 electrons. This is, it's going to be stable. It has 2 in this ring right here. So it's all good to go. All right, let's check this slide out from class. All right. So everything's made of matter. Matter's made of atoms. Elements are made of one type of atom. So that's what I just went over with the previous slide. And here is the basis for water. So you can see hydrogen has 1 proton and 1 electron. It has no neutrons. Its number is 1 on the periodic table. Oxygen's number on the periodic table is 8. It's going to have 8 protons, 8 neutrons, and 8 electrons. There's the first ring holds 2 electrons, and the second ring can hold, or what we actually call an energy level, can hold up to 8. So if this one holds 2, you can see that there are 6 in this ring here. That's going to be important for later on. All right. Once again, you can see up here that this will not be on the assessment. It's just to help you learn about it. So electrons determine the chemical behavior of an atom or who it wants to bond with. And it depends on the number of electrons in the outermost shell. And there's something called the octet rule here, where atoms are satisfied if they have 8 in their outer shell for the most part. And so here with sodium and chlorine, if you look them up, chlorine has 7 in its outer shell and sodium has 1 in its outer shell. Sodium will lose that electron or transfer it to chlorine. That satisfies chlorine for the octet rule because it will now have 8 in the outer shell, and sodium is satisfied as well because it lost 1, so its outer shell, the one below that, was full. All right, that's called an ionic bond. Let's check out covalent bonds, and that's what I had as question 3 on the study guide. Ionic bonds will not be on your test. So here we have hydrogen, and then we have a picture of a water molecule. And in this example, hydrogen has bonded with itself to become H2. It is now sharing electrons, this hydrogen sharing with this hydrogen, and that gives it 2 electrons in this level. They're sharing them. Both atoms are happy. They're all good. They've fulfilled their shell right here. Let's look at this one. Oxygen forms with 2 hydrogens to make H2O, which you all know. And now we have a water molecule. The H's are satisfied because they have 2 in that first energy level. They have 2 electrons. And the oxygen is satisfied too because it has 1, 2, 3, 4, 5, 6, 7, 8 in that second energy level. So everybody has the right number of electrons. All right, that would be a covalent bond. It's very strong. That is an intramolecular force. They are sharing within the molecule. Now, water is going to make life possible, and it's going to be a polar molecule. So let's check this out. Can you describe why it's a polar molecule? So go ahead and stop the video. All right, let's check it out. Here we go. Let's make sure I got everything from this slide. Due to the oxygen having more protons than the hydrogens, the oxygen will hold the electrons more often than the hydrogens do. It could be like a big kid and a little kid in tug of war. The big kid will hold or be able to pull on the little kid more often. And so the oxygen will be relatively negative, as represented here, whereas the hydrogens will be relatively positive. The fancy word for this is called electronegativity, that you'll learn about in chemistry, the affinity for electrons for an element. So oxygen in a water molecule will be relatively negative, and the hydrogens will be relatively positive. This is important because now we have established that water is a polar molecule, it has poles. It has negative and positive poles. Well, guess what? Those negative and positive poles, this is just a repeat of what I just said, those negative and positive poles will now like to bond to each other. So a positive hydrogen, right here, relatively positive, will form a hydrogen bond with a relatively negative oxygen here. This is a weaker bond, and it's represented with dashes, but it's very, very important. The bond between the oxygen and the hydrogens, that covalent bond, is stronger than the hydrogen bond between the 2 water molecules. Here you can see the positive hydrogen connected to a negative oxygen. Another positive hydrogen bonded to this negative oxygen. Here, this positive hydrogen bonded to a negative oxygen. This doesn't just happen in water. It can happen in DNA and other substances as well. It will always involve a hydrogen, and it's a hydrogen bonding to something that is negative. Opposites attract. All right, just like we saw in the Camp Sticks performance. Here is the covalent bond between the oxygen and the hydrogen. Here's the hydrogen bond between the hydrogen and the oxygen. Okay, this is going to lead to what we call emergent properties. And this is a fascinating concept. And so if you think about CISA-winning football teams here at our school, they can only do so much with just 1 player. They can only do so much with 2 players. But you give them 7 on a side or 11 on a side, they're able to construct plays and pass the ball and do items that they can't do on their own. So here, the emergent property of hydrogen bonding hydrogen bonding leads to emergent properties for water. And so let's go through them. Due to hydrogen bonding, we can have transpiration, water going up a tree. Water is able to absorb lots of heat, right? So as you heat up water to make spaghetti on your stove, it takes a long time to get that water boiling. Water helps to cool us down. We are 60% water, if not more. So it's the medium in which our cells live. Water is less dense as a solid, so therefore ice will float, and it can dissolve lots of items. So let's dive in a bit more about these emergent properties of water that contribute to life. These are due to the hydrogen bonds. All right. Cohesion would be like water sticking to itself. So here you can see that they're cohering to themselves as this little droplet on my finger, and an adhesion is this sticking to my finger. So I just remember like a couple, and adhesion, and it adheres. So why is that important? Well, it leads to what's called transpiration. If trees had to expend energy to move water up to their leaves, that would cost them tons of energy. We wouldn't have really tall trees or tall plants. But because water can adhere to the xylem wall and cohere to itself, it's able to get pulled up like a chain up a plant. When water evaporates out of the stomata in a leaf, it pulls up the next water molecule. And this works to get water all the way up a tree. We modeled that in class with the capillary tubes as well. All right, as water evaporates, it's taking away heat, right? It's evaporating at the surface. So when we sweat or when plants release water vapor, it helps to cool them down. It's taking away the most energetic molecules. It takes longer to heat up water than other substances. So if you were to put on your stove a pot of water and then a pot of alcohol or another substance, the alcohol would heat up more quickly. The water has to break all those hydrogen bonds before it starts to boil. Because it takes a long time to heat up, this helps lakes and oceans to be able to absorb large amounts of heat. So if you've ever gone into a lake or the ocean at the beginning of summertime, it's cold. Well, that would be in the U.S., not as much here in Oman. But then, they're not going to get cold. Once they get warm by the end of summer, they're not going to cool down with just one night. So they hold on to heat. They can absorb a large amount of heat. And that's helpful for us, right? This morning, I went out running, and I ran my usual 7 kilometers at a slow pace. And I was rocking like 165 beats per minute with my heart rate because it's just so hot right here in Oman. And so I would have died if I didn't have a lot of water in my body because the water helped to moderate my body temperature. Sorry, that was a non sequitur. All right, water expands upon freezing. So here you got to model this with those water kits. And so in the liquid water, the hydrogen bonds are constantly breaking and reforming, breaking and reforming. And in ice, they form a lattice structure. And so they're more spaced out. Density is mass divided by volume. So if in ice, if they're more spaced out, volume is going up. So mass divided by volume, it would be less dense, right? It would be the same mass, but then the volume would be larger with the ice. So this is really good for us, right? Because it allows ice to float on water. So when lakes freeze or when the ocean freezes, it freezes on top and then there's water below. If it froze from the bottom up, then all the fish would die in the ocean, right? And it would quickly freeze over because they'd be constantly exposed to the colder air temperature. Water is also very good at dissolving substances. So here is an example of salt. And so notice how those positive hydrogens are going to cluster around the negative chlorine atom, and the negative oxygens are going to cluster around the positive sodium ion. This also works with sugar, right? You put sugar in your coffee or your tea, and it dissolves. It doesn't break the actual bonds between, say, the sucrose molecule or the glucose molecule, but it pulls them apart from the big crystalline structure that they were in. All right. And then here's my daughter climbing a tree that I put in here to represent transpiration. That was atoms, water, and life. Remember atoms and that the physical chemistry won't be on your assessment, but water will be. Okay, let's dive into carbs, lipids, proteins, and nucleic acids. So how are they assembled and disassembled? What are the functions of each group, and how does the structure of each group lead to its function? So when you take chemistry next year, we often say that the structure of a molecule leads to its function. So how it's set up leads to its function. That makes sense. If you think about, let's go through some examples. So here are my kids at Legoland Dubai. This was not this spring break, but the one before that. We had a ton of fun, and we were making some Legos, right? And so each Lego on its own would be a monomer, and putting together makes a polymer. The structure of this leads to its function of being a toy for my kid and a fun cafe that she can play games with. So here we go. Let's dive into polymerization. A polymer, that prefix means many. So it'd be many monomers put together. A monomer is that building block. So it'd be like one Lego, and then putting many Legos together makes the bigger one. This is done through a process called dehydration synthesis. In dehydration synthesis, we're going to dehydrate or remove a water molecule as we synthesize the two monomers together. Let's watch this happen for glucose. We got to model this with some paper models in class. Here we're going to have the two join together on this oxygen. We'll remove this H and this OH that removes a water molecule. And now we have dehydrated and we have formed a bond on that oxygen. This bond is very strong. It is a covalent bond. They are joined together, and we're not going to break this apart unless we use an enzyme. So we have dehydrated. We have removed one water molecule and then formed a bond here on this oxygen. Oxygen has 6 valence electrons, so by forming a bond with a carbon here and a carbon here, oxygen is satisfied. All right. In hydrolysis, I'm sorry, I was talking about dehydration synthesis, and I'd gone on to the next slide. In hydrolysis, we insert a water molecule with the help of an enzyme to break apart a polymer. So let's see it in action. Here, it's not, okay. I'm going to have to pause for a second and edit the slide. Okay, through the magic of the internet there, I now have a picture of hydrolysis. So here is a sucrose molecule, 2 monomers put together, and with the help of an enzyme and energy, the enzyme called sucrase, breaking apart that sugar you might have eaten for breakfast, it's going to insert a water molecule on that oxygen. This oxygen, that was a strong bond, it is now broken, and it inserts an OH here and an H here. It's not important that you remember where the OH or the H goes. What's important is that you know that water is used to split the two molecules. Hydro meaning water and lysis meaning to break. If you want to check it out, you can see that oxygen has 2 bonds, one with the H and one with a carbon right here. This is tricky, but if there's no letter, it's understood to be a carbon. This one is bonded with an H. This oxygen is bonded with an H and with a carbon. So they're both fulfilled. And now we have glucose and fructose. So we've gone from a bigger molecule to a smaller one. Why is that important? Well, glucose can enter the cellular respiration pathway and then be oxidized to make energy. All right, I'm back. So let's move on. So we just did dehydration synthesis and we did hydrolysis, so the formation of polymers from monomers and then breaking down a polymer into monomers. So for instance, like today, I just had lunch duty, that's where I just came from. I watched some students eat some sugar, right? And they're going to break that down into constituent monomers so that they can do other stuff. All right, now let's get into the major biological macromolecules. Those are carbs, lipids, proteins, and nucleic acids. So can you talk about what the function of a carbohydrate is? And then how does the structure of this contribute to its function? Once again, just like we said with water, the structure of the water molecule contributes to its function, and many other chemicals, same thing for a carbohydrate. It's a hard question. Let's break it down a little bit. Do you remember what the ratio of elements in a carbohydrate are? Well, if you got C6H12O6, which is glucose, you might remember that from our second unit, but usually 1 to 2 to 1 for carbon, hydrogen, and oxygen. So 1 carbon, 2 hydrogens, 1 oxygen. There are many different types of carbohydrates. For example, starch and cellulose. There's amylose, there's big sugars, there's branch sugars, there's glycogen, etc. And learning about those is more of an AP bio thing. We're We didn't have time in this unit to really dive into that. The reason we went over the chemistry of life is because they make up the parts of a cell. So an example would be starch and cellulose are made of glucose molecules in different arrangements. So here would be starch. Notice how all of them have the CH2OH on top. And then look at this. In cellulose, it's flipped, right? And so they're made in a different arrangement. These are going to be called beta glucose monomers, and these are alpha. We can eat the first one. I love it, man. I love French fries. So we eat the starch, but we can't eat cellulose. Cellulose would be like tree bark. That's not something we could do. Some organisms that can might be termites, and they have bacteria that they have a symbiotic nature with that help to break these down. All right, so like we said, 1 to 2 to 1 ratio. The polymers will actually not be in quite a 1 to 2 to 1 ratio because you remove a water molecule from dehydration synthesis. For example, the formula for sucrose should be C12, and then we remove one H2O. So instead of H24, it should be H22. And instead of O12, it should be O11. All right, they provide quick energy. A trick for this is that they often end in OSE, like sucrose, maltose, fructose, glucose, etc., and they're made through the process of photosynthesis that we studied in depth. One sugar is called a monosaccharide. It means one sugar. So that's what saccharide means right there. Examples would be glucose and fructose. Disaccharides, di means two. Saccharide still means sugar, so this would be two monosaccharides joined together, like maltose or sucrose, and that's through dehydration synthesis again. Polysaccharides would be many of them put together, and they could be storage ones, such as starch and glycogen, or structural ones like cellulose. All right, let's move on. Oops. All right, their function is quick energy. Their structure is in a 1 to 2 to 1 ratio. In the watery environment of our body or in water, they usually are like a ring-shaped structure that can be broken apart. All right, we started with carbohydrates, and we did that very quickly, but now we can tie it all together with lipids. Are we ready? What are lipids? What are the functions of the following lipids? And what structures lead to the functions listed above? Again, you can pause the video there for a second. All right, when we think about lipids, we should think about fats and oils. All right, also included there are waxes and steroids, but basically things that don't dissolve in water. When you pour oil on water, it floats on top, and that is because it is non-polar. These long chains of carbons and hydrogens are non-polar. They do not interact well with water, and so they will form their own droplets on top of the water, and they move away from it. Fats are really important. They insulate us. They give us warmth. They provide energy, and they serve as our cell membranes. And we've done a lot with the cell membrane throughout this unit and how cell membrane provides for compartmentalization within a cell. Here's what's so interesting about structure leading to function. Lipids are also made of carbon, hydrogen, and oxygen, just like carbohydrates, but they're in a different ratio, in a different shape. And so here, there's going to be relatively more carbons and hydrogens in a lipid than there will be in a sugar. Lipids or fats store more energy than carbohydrates per gram. So that's why if you eat a bunch of carbohydrates, you don't burn them all up for energy. Your body will turn them into fats because it's a more efficient storage mechanism of that energy. So once again, structure leading to function. All right. Here, we could dive a little bit more into fats just for you in case you're interested. Here would be a glycerol molecule. A glycerol molecule is the backbone of a fat, and so I'm not going to ask you that on the test. That's more like AP stuff. But what happens is there's a dehydration synthesis reaction between the glycerol and the fatty acid here. It's called an ester bond or an ester linkage. And then you'll have the 3 fatty acid tails, right? If there's a double bond, then there will be a kink like this, and that's called an unsaturated fatty acid. If there's no double bonds, they're like this. You might say, why is that important, Mr. Gibney? Once again, it helps to show the theme of structure leading to function. These unsaturated fatty acids will be liquid at room temperature, whereas saturated fatty acids will be solids at room temperature. This would be like butter, and this would be like olive oil. Our cellular membranes are a combination of saturated fatty acids and unsaturated fatty acids to provide for the right fluidity in the cell membrane. Let's see dehydration synthesis for lipids in action. So there's an enzyme at play. The water molecule is going to be removed. This is going to now bond together, and we're good to go. We can also witness hydrolysis. So here comes the enzyme to hydrolyze it. We're going to insert the water molecule. The H goes here, the OH goes here. Bam, we've now broken apart the fat. So let's say that you want to burn some fats. You first of all need to go exercise. You need to raise your heart rate. You're going to burn your carbohydrates. After you've burned through those, you'll then start to burn fats. So you'll actually be doing this hydrolysis reaction here, and then these will enter the cellular respiration pathway. All right. Structure leading to function, right? The structure of a molecule leads to its function. Well, here, let's look at this phospholipid that's going to make the phospholipid bilayer. The big thing here, this is a little complicated chemistry, is that the head likes water. It is hydrophilic. You can remember that L in it for loving water, philic, hydrophilic. It likes water. The tails are going to be hydrophobic. They do not like water. They repel water. They are nonpolar. The head is polar. So when you put them together, when you make a bunch of them, they will naturally form this lipid bilayer. No energy required. The phosphates, or the heads, will face towards the water, and the tails will face on the inside to maximize their distance away from the watery environment. So here would be the non-polar inside and the polar heads. Steroids is not something we went over in this class, but they also count as lipids because they are non-polar and hydrophobic as well. These are things like testosterone, estrogen, cholesterol. Okay, so lipids are another great example of how structure leads to function. Go back through the video, check it out, see how the structure of different types of lipids leads to their functions. Let's jump into protein. So we were up in Jebel Akhdar the other weekend, and we went to this breakfast buffet at the Anatar. My goodness, it was just fantastic, right? And so there we ate some protein. Can you ask yourself, what are the functions of proteins? What's the structure of a protein, and how does this lead to its function? You might think back to when we went out in the hallway and we did the protein party, where you had different R groups on you and you all interacted. All right. Proteins are made of carbon, hydrogen, and oxygen, which carbohydrates and lipids are, and then the addition of nitrogen and sometimes sulfur as well. So CHON. What happens is that there are amino acids are the monomer of a protein. They are the building block. They are the single Lego, and they combine together to make the polymer. Proteins do everything. You're looking at me, you're looking at proteins. They work as enzymes, they build muscles and bones. They help to fight infections as antibodies. Here is the primary structure of a protein. That's going to be the order of the amino acids. And you might remember our codon chart where we use DNA to figure out the RNA to figure out what amino acids are going to be coded for in a gene. And so usually to start with AUG, which is going to code for methionine, right? Amino acids have directionality. They always start with the amino group there on the first part of the protein. And then there will be what's called a central carbon. That central carbon is going to be bonded to a hydrogen and an R group. The R group differs between the 20 amino acids. It will then be joined to a carboxyl group, which is COOH. This group is called the amino one, and this is a base, and this is the carboxyl group, and this is acidic. So this is an amino acid. All right. I'm not going to ask you anything that in-depth on your test, but it's just good to check it out. All right, here we go. Here's some amino acids and their structures. So notice how lysine has this long chain, glutamine has this chain, aspartate has a much smaller one, alanine is much smaller. And so this is just showing differences in the R groups. These R groups will interact with each other later on. We'll get into that. Here's where that sulfur comes from. It's in cysteine and methionine. All right, we're bringing it up again. Dehydration synthesis. Here we go. We kick out that water molecule and we bond together the two amino acids to make a polypeptide chain, many peptide bonds. Here it is in action. So here goes the carboxyl group and the amino group. They're going to remove that water molecule and form this bond. This is a covalent bond. This is the sharing of electrons. We call that a peptide bond. Here's an example of hydrolysis, and we break it apart, right? So if you ate some protein for, I saw some students eating protein for lunch, or like I said with my kids right there, they are hydrolysizing it. They are breaking it apart so that then they can have those monomers and then build them together in their body for the proteins that they want. All right, this is AP level, so this is outside of our course, but in the secondary structure, they start to form these helices or beta-pleated sheets. A guy Linus Pauling figured this out while he was sick one day. He's home sick, and he just sketched it out. Tertiary structure is what we did in the hallway. So that's the R groups. Some R groups will want to be on the outside of the protein, and some R groups will want to be on the inside. Sorry, just had to answer a student question there. So the R groups, they will start interacting with each other. Some of them that are polar will go towards the outside of the protein, right? Because they like water. The non-polar R groups will go towards the inside. That gives a protein its shape. And in quaternary structures, just if you put together multiple proteins. All right, and then proteins can act as enzymes. We'll do that after nucleic acids. All right. We've already spent a big chunk of time on DNA. That was the first test of this semester. I know it was challenging. You did a great job. I was quite proud of you. So let's just do a quick review on structure and function of DNA. All right. Hopefully you remember that the function of DNA is to serve as hereditary information, right? It's how we pass down our hereditary information. The structure of it is going to be a sugar phosphate backbone with a nitrogenous base. That's going to form hydrogen bonds with another nitrogenous base that is attached to a sugar phosphate backbone. This is what we call a nucleotide with the phosphate, the sugar, and the nitrogenous base right here. So that would be one nucleotide. That would be the monomer. Here's another nucleotide. This picture is good because you can see that there's directionality in DNA as well. This one is going in one direction, and this one's going in another direction. So they run kind of like salt and caboose highway. They run opposite of each other, kind of like this. All right. DNA has A's and T's that pair together and G's and C's. And then when we make RNA instead of T, there's going to be U. RNA will be single-stranded and has a different sugar. It has to help differentiate it. All right. Let's move on. Just like all the other things, right? We said that they are linked together by dehydration synthesis. Let's see if I have a picture of it. No, I don't. Dehydration synthesis, one water molecule is going to be removed, and then the next nucleotide will be added. The interesting thing about DNA is that it can only occur in one direction. It has to occur what's called in the 3' direction. And it's just because that's where they're able to remove an OH to make a water molecule. You can't remove it off of the phosphate group, off of this P here. All right, let's move on. Enzymes. So enzymes are proteins. And so that's why I included them in this unit. And they're absolutely critical to our body. So we did a lab, and the lab was hydrogen peroxide and catalase. Catalase is an enzyme that breaks down hydrogen peroxide into water and oxygen. Hydrogen peroxide is H2O2, and then with the addition of catalase, it's broken up into water and oxygen. So can you describe how enzymes speed up chemical reactions, the lock and key theory, and describe how temperature and pH affect them, and substrate concentration and enzyme concentration? And you should be able to discuss the lab that we did in class. All right, let's dive in. Enzymes are proteins, like we said, and they speed up chemical reactions. Without them, we would die. They lower the amount of activation energy needed for this reaction to happen. So let's look right here. Here is a graph showing reactants and products. There has to be this much energy for the reaction to take place. It'd be like getting started on a bike. You got to push down and get going before you then might be able to coast down a hill or go somewhere, right? You got to give it an initial push to get going. Well, the enzymes make it easier. They make it easier to do. Maybe you're able to get a little kick off of the ground, etc. Here's another example of it. So this is reducing the amount of energy needed for the reaction to occur with the enzyme. Enzymes are everywhere, right? They help us build molecules. They help us to break us down molecules. They are what do dehydration synthesis and hydrolysis. They help to speed up reactions. Each of your cells has hundreds of thousands of enzymes. Let's check them out. They're proteins. They're specific to their job. It needs to be the right shape to do its job. And they're named for the reaction that they help. For example, sucrase is an enzyme that breaks down sucrose. Proteases break down proteins. Lipases break down lipids. They often end in ASC. Here is an example of enzyme in action with all these vocab words. So here we go. Here is the enzyme. It's purple. It has an active site. The active site is where the substrate binds to the enzyme. In this example, the substrate is being broken apart. It could be put together. The substrate bonds tightly at the active site. The enzyme then catalyzes the reaction, and in this instance, it cleaved the bond right here and spit out the product. The enzyme can be used again and again and again. All right, it can be keep using again and again and again. They're specific. This wouldn't work on, say, a reaction that was dealing with two triangles, right? It doesn't work. It has to be the correct shape here. And I think that's it. Let's go on. It's the shape that matters, and that's why we call it the lock and key model. So, like, your key works on your house. It doesn't work on my house. Doesn't work on a friend's house. Your key works on your house. So each enzyme is specific to the actual reaction it does. I think we went over the vocabulary, right? The enzyme is the protein. The substrate is the molecule that the enzyme works on. The product is what comes out of it. And the active site is the part of the enzyme that the substrate fits into. So let's do the example of our lab with that, right? We had hydrogen peroxide. The hydrogen peroxide is the substrate. Then we have the enzyme. The enzyme is called catalase. That would be this. This would be catalase, right? The H2O2 fits into the active site. It is then cleaved, and it makes H2O and oxygen. All right, I know it's not quite balanced. That's okay. And those would be the products. Here was your lab, and so you saw that the liver had quite a reaction with the hydrogen peroxide. But and then we saw that all living things had catalase in them as well. But if you boiled it, it didn't work as well. If you cooled it down, it didn't work as well. If you put it in vinegar, or if you put it in baking soda, it didn't work as well. And that shows you that there are things that affect the activity or the rate that an enzyme works. So here we have an example of pH. This enzyme works best at a pH of 8. Enzymes often work best at neutral pH because that's the majority of things in our body, but not always. For example, an enzyme that works best in your stomach might be an enzyme that works in an acidic pH level. An enzyme that works well in your intestine might work well at a basic pH level. Temperature affected it, so if we boiled it, we actually denatured the protein. We cooked it, and it no longer had the shape that it could match up with hydrogen peroxide, and it didn't work at all. If it's too cold, then the molecules just combine together less. All right, and here's what a denatured protein looks like. Let me get the substrate concentration and enzyme concentration up here. All right, we're almost at the end here. So here is a good example of heating up an enzyme and it becomes denatured. Notice how this enzyme can no longer fit with this substrate. It will not work anymore. Here's another picture of it. So here's an enzyme, amylase. It's going to break down starch. The active site will work upon the substrate, but once it's denatured, it no longer works. There could be things like inhibitors and poisons that also affect enzymes. Want to learn more about that? Take AP Bio. It'll be fun. All right, let's look at this graph. So I would give you a graph. I wouldn't expect you to memorize what happens with substrate concentration and enzyme reaction rate. But let's pretend that these purple ones are sucrases, right? And then this substrate is sucrose. It's the sugar. Well, as we add more sugar, right, more of these enzymes will their active sites be occupied and will be forming more product of glucose and fructose. So the reaction rate increases. But at some point, we will saturate all the sucrase enzymes. There will be no free ones. So if we add more sugar, it won't speed up anymore. Right? It's gone, it's gotten better, it's gotten faster, and now it's just kind of leveled off. It's kind of like my ability to play football. I got to Tasem, I didn't know anything about it, I got better, and now I've just kind of reached my natural limit. That was maybe not the best analogy. So here we go, as the amount of substrate goes up, the reaction rate goes up until it levels off because all the enzymes are saturated. You can pause the video and check out this slide. This just kind of summarizes it as well. And now let's do it for enzyme concentration. So here, as long as you have enough substrate, as you add more enzyme, the reaction rate will increase. It's because now they'll collide more frequently with the substrate. Eventually, the substrate will become the limiting factor. You only have but so much sugar inside of you, and then not all the enzyme molecules can find a substrate, and it will also level off. All right, students, that is it for the chemistry of life. The video is a little disjointed, but I hope you found it helpful. Can't wait to see you crush it. Have a good one. Hit me up with any questions.